Hello, my name is Alicja Chmielewska and I'm a molecular virologist. I work in the laboratory of virus molecular biology at the Intercollegiate Faculty of Biotechnology of University of Gdańsk and Medical University of Gdańsk. And today I want to tell you about viruses and vaccines. I will explain what they are. I will also concentrate obviously on COVID-19. And even if this is a lecture on molecular virology, I will try to use really simple terms to describe this matter so that everybody can understand. So what is molecular virology? So virology is a broad field, and for example, a clinical virology concerns more the disease itself and the symptoms of the disease. And for example, epidemiology is more about how the viruses spread in the population, whereas molecular virology concerns mainly the viral particles, so their structure and how they enter the cells, how they replicate in the cells, and also we try to discover new therapeutic strategies against viruses, like for example new vaccines and new drugs. So in our everyday work we use obviously different kinds of microscope materials, but what we do really a lot is to make cell cultures. So we culture cells in special dishes, and almost like all experiments that we make with viruses we make in the cells, and because the viruses, they amplify in the living cells, so in the cells that build our organisms, and I will explain more about it in the next slides. So viruses are everywhere, and they infect all the living organisms, not only humans and animals, but really all organisms that are on our globe. So they can infect plants, they can infect insects, they can infect even microorganisms. And here on this slide you can see infection of bacterial cell by viruses. On the left you can see visualization, and on the right you can see the real picture made by electron microscopy, where you can see many viral particles that are invading bacterial cells. So even the bacteria suffer from viruses. And here on this slide you can see what is the size of the virus, and what we need to see viral particles. So first you can see the objects that we can see with naked eye. Then in the middle you can see the objects that we can see with light microscopy. So for example, animal cells and plant cells, and also bacteria. And viruses are much smaller than bacteria, and they are so small, they are about, like average virus is about 100 nanometer, that we cannot see them with light microscopy. So we need to use special equipment which is called electron microscope to see viruses. So here you can see some pictures of the viruses from electron microscopy. On the left, it's Ebola virus with its characteristic shape. In the middle, there is a picture of particles of SARS-CoV-2, of the new coronavirus, and you can see its characteristic corona outside of the particle that gave the name to coronaviruses. And on the right, you can see some viral particles of human herpes virus. So these pictures are black and white. If you can see some colorful pictures in the internet, it means that they are colored, but like normally these pictures are always black and white. So what is a virus? In very simple terms, we can say that the virus consists of genetic material and the code. And its genetic material is very important because it is a recipe for a virus. And the code is there to protect this recipe and also to enable the virus to enter the cells, because it is the cell that needs the recipe to produce more viruses. And how is it happening? So the cell in the organism has on its surface some proteins called receptors, and they have various functions in the organism. And the virus also has some proteins on its surface. They are called just surface proteins. And if the viral proteins on the surface match with the cellular proteins on the cell surface, like a key to the lock, then the virus can enter the cell. In the graph number 2, you have depicted how the virus entered the cell, and then the genetic material, so the recipe for the virus, is liberated from the code of the virus, and then on the basis of this recipe, the cell can produce all the elements that are necessary to build a virus particle. So, for example, it has to produce more copies of genetic material, so more recipes that will enter the ready particles. Then the cell produces more codes, the white structures here, more codes that will protect these recipes from physical-chemical conditions, and also they will make the structure of the virus. And also the cell produces more of the surface proteins that are depicted here as blue circles, because then these new particles will also be able to enter new cells. And then all these elements are brought together to construct viral particles which escape from the cell, and sometimes they break the cell, and they are ready to infect new neighboring cells. So what is very important from this slide, it is that it is the cell that produces new viral particles on the basis of the recipe, and the recipe is the genetic material of the virus. And that is why we, as molecular virologists, need to culture cells, because the whole cycle of the virus happens in the cell. So if we want to test any antiviral strategy, like we want to test antibodies against the virus or some drug against the virus, we need to apply it to the virus in cell cultures, so we are able to see if it stopped the virus, for example, from entering the cell or from replicating in the cell. So that is why we work with the cell cultures. So here I would like to show you another way to see the virus. So I already showed you how we can see the virus with electron microscopy, and with electron microscopy we can see very tiny objects like nanometer, like the size of the nanometers, and we can see viruses in black and white, and we can see directly viral particles. But here I want to show you some method. It's not like direct observation, but we can also observe viruses indirectly, because we can observe infected cells. So if we add virus to the cells that are cultured in a dish, then we can see some difference in these cells if the virus enters and if the viral proteins are produced. So on the left you have the picture of the cells that were infected with tick-borne encephalitis virus. So this is a dangerous virus transmitted by ticks that is very common in Central, Eastern and Northern Europe, and also Northern Asia. And in the middle you can see some cells that are darker, and also they have some different shape than surrounding cells. So these are the cells that are already dead because they have been infected with the virus amplified and the cells died. And this is called a viral focus, and we can observe it by light microscopy. So this is the effect of the cells that was that resulted from virus infection. Then below you can see some cells that are fluorescent with red color, and also the nuclei of the cells. So the centers of the cells, the nuclei are blue. So we have methods of molecular biology that enable us to stain the cells that have been infected in such a way that they are fluorescent, and we can observe them by fluorescent microscopy. On the right, in the top, you can see cells that were infected with adenovirus, to which by different methods of genetic engineering I inserted a gene that codes for green fluorescent protein. So it means that this adenovirus delivers recipe for green fluorescent protein into the cells. So the cells that have been infected, they produce green fluorescent protein, and we can observe it by fluorescence microscopy. And this is quite interesting because this adenovirus encoding for this fluorescent protein is actually very similar to one of the anti-COVID-19 vaccine. This is Oxford vaccine, which also contains adenovirus, which also carries a foreign gene. But here we have a gene of green fluorescent protein, and in the vaccine we have a gene of SARS-CoV-2 virus, which then is produced in the cells and becomes a vaccine. And then in the right panel beneath, you can see insect cells that have been infected with baculovirus, which is an insect virus, and the infected cells are dyed with red, so you can see that also insect viruses infect their cells. This is how indirectly we can see viral infection using cell cultures. So to sum up, what is a virus? So we can call it an intracellular parasite, because it doesn't have its own metabolism. It is able to perform processes only in a living cell. So we say that viruses are at the border of living and non-living matter, because outside of living cells they resemble non-living matter, but inside living cells they resemble living organism to some point. They are smaller than bacteria and they have no cellular structure. And we can define viruses as genetic material, recipe, that is surrounded by protein shell and also eventually lipid envelope. They are not sensitive to antibiotics. So antibiotics are specific drugs against bacteria, but not viruses. So now I will start the part of the lecture that concerns more SARS-CoV-2 and COVID-19. And first, to clarify, I just wanted to mention that SARS-CoV-2 is abbreviation from Severe Acute Respiratory Syndrome Coronavirus 2, and COVID-19 is abbreviation from Coronavirus Disease from 2019. So it is very important that this is not the same. So like SARS-CoV-2 is a virus and COVID-19 is a disease that is caused by the virus. And SARS-CoV-2 has similar structure to the general virus structure that I already showed you. So inside the virus particle there is RNA. RNA is its genetic material. So this is the recipe of the virus. So it contains all the information about all the proteins that make the structure of the virus. Then outside genetic material there is the protein shell that should protect the genetic material. Then there is also a lipid envelope, because there are some viruses that are enveloped and some that are not. This is an enveloped virus. And then in this lipid envelope there are proteins called spike proteins, and these are the proteins that bind to cellular receptors. So these are those proteins that can serve as a key that binds to the lock to enter into the cells. So, as I showed you, SARS-CoV-2 viral particle contains lipid envelope, and presence or absence of lipid envelope is quite important for us and for our daily life with the virus, because the presence of lipid envelope determines resistance of the virus in various conditions. So sometimes I ask my students question what they think if the virus that has a lipid envelope, if it is more resistant or less resistant to various physical chemical conditions that non-enveloped virus. So it is tricky because first it seems that this lipid envelope is something additional, so that is additional protection, but it is not like this, because the surface proteins, the keys, are embedded in the lipid envelope, so it means that this lipid envelope is essential for the virus. If we destroy lipid envelope, then we destroy the virus, because it loses the key that enable it to bind to the lock and to enter the cells. And because lipids are quite sensitive to dissolving in many reagents like detergents and ethanol, also enveloped viruses are not so resistant, and we can destroy these viruses with detergents and alcohols. And that is why we are advised to use soap to wash hands, and also to use alcohol disinfectants when we are outside the house, so that we do not only remove the dirt from our hands, but also we just kill the virus by destroying its structure, and more specifically by destroying its lipid envelope. And now from virus molecular structure, we will move to vaccines. So vaccines are present in our life already for a long time. You can see here some old painting with magicians injecting vaccines. So the first attempts of vaccination were as early as the year 1000, and the first trial was in China against smallpox, and it was called variolation. It was vaccination against smallpox with the use of infectious material from patients who had mild version of the disease. So basically they took some material from infected patients, like infectious materials from the pustules, which are the changes that develop on the skin of the people that suffer from smallpox, which is very dangerous disease. And they inject this infectious material into small lesions on the skin, and then the patient who received such immunization developed mild disease, and it didn't take long, like the disease was 1 week, 2 weeks, and it was not fatal. So it was much safer than the real disease that was very often fatal, and it was used in China, in other countries of Asia. It was also used in Africa. And in the 18th century, Edward Jenner in Europe used cowpox infection to protect against smallpox. So he noticed that the women that were milking cows, that they very often had some changes on the skin on their hands, like pustules on their hands, but they very rarely contracted smallpox. So he started to experiment, and it turned out that infection with cowpox, the virus that infects cows and is very similar to smallpox virus, that infection with cowpox is protecting against future infections with smallpox that is much more dangerous. So it was used as a first, let's say, modern vaccine. Then in 19th century, Louis Pasteur successfully tested a vaccine against rabies. And then in the 50s of 20th century, the first polio vaccines were administered to children. And I just want to mention that polio epidemic was a very dangerous epidemic and very frightening one. The polio virus is very dangerous for children, and children who suffer from this disease very often have long side effects, and they can be disabled for the rest of their lives. And parents were really afraid of their disease. They were afraid of letting the children out of the house during the epidemic. So there was really a lot of scare in that time. So this vaccine was welcomed with really relief and hope for better health of the children. Then in 1977, the last case of smallpox was reported. So now smallpox is eradicated. It means that there are no longer any smallpox cases in the world thanks to vaccination. And also, what can we say nowadays is that number of deaths from measles, which is also a very dangerous disease, is reduced by 80%. Then maternal and neonatal deaths from tetanus, they are also nearly eliminated, and polio is also nearly eradicated globally. But in 1990s started this public scare about a link between vaccines and autism, and this lecture is not about this, but I just want to mention that there is no scientific proven data that would confirm any link between vaccines and autism. So any suggestions like this are not scientifically correct. So here, just to show you what is the impact of vaccines in our world. So here you can see the cases of smallpox in children, and as I told you before, smallpox has been eradicated thanks to mass vaccinations. On the right you have the picture that allegedly shows two brothers, and one of them was vaccinated and the other was not. Also here you can see some pictures of children suffering from viral infectious diseases that now are not a problem anymore thanks to vaccinations. So on the left you have the measles, down mumps, and on the right there are children that are respirating thanks to some special equipment called iron lungs that was necessary because their respirating muscles were not functioning properly. But as vaccine use is very long, the same is for anti-vaccination movements. So in this old painting you can see the reflection of the fears against vaccination with cowpox, a virus from cows that was to protect from smallpox. So according to the author, people vaccinated with cowpox preparation would develop some cow organs and would transform into cows. So the fear against vaccines is not something new. It had different forms throughout history, but it was always present in the society. So what are the vaccines? In simple terms, vaccines mimic natural infection, and this allows our organism to develop immunity. So it is something similar to a real virus, so that our organism can sense it, learn it, and then develop immunity against the virus itself. And classical vaccines can be segregated into different types. Classical vaccines are the vaccines that we all used, we all have been injected in our childhood. So there are these all vaccines against mumps, rubella, measles, hepatitis B, and so on. So one of the type of classical vaccine is inactivated virus, which means that it is like killed virus. So normally a virus, when it enters our cells, as I showed you, it is able to enter the cells and amplify. Inactivated virus looks like a virus, it has the same structure, but it is not functional and it cannot develop any infection. So if you, for example, boil an egg for the breakfast, you can see the difference between the raw egg and the boiled egg, and you know that there will be no chicken from the boiled egg. So also the virus we can inactivate with the heat, and all the proteins that make the structure of the virus become denatured, so they are inactivated. They are not functional anymore. We can also use other procedures to inactivate the virus. Then another form of the vaccine is attenuated virus, which is a living virus, which is weaker, much weaker than the normal virus. So it is a virus that can propagate, can replicate in our cells, but in a very non-optimal way, so that our immune system can very easily get rid of this virus, can fight it very easily. And another kind of a classical vaccine is a vaccine that consists just of a part of the virus. So it is not the whole virus. It can be just one protein, for example, a surface protein of the virus, the key that is used to enter the cells. Or it can consist of all the viral proteins, but without genetic material, so without the recipe. So if our immune system sees a part of the virus or the whole virus without the genetic material, our organism, our immune system can remember the parts of the proteins to fight against, but as there is no recipe, our cells will not propagate this virus, will not produce any new offspring of the virus. All these vaccine types that I showed you, they are made with the use of the infectious virus, because if we want to have killed virus or weakened virus or part of the virus also most often, we need to have the virus first. So we need the virus in order to kill it or to weaken it. And to have the virus, we have to amplify it. And as I showed you, the virus amplifies only in living cells. So in factories producing such vaccines, virus amplifies in cell cultures, as I showed you before, in special dishes, or it can be also amplified in chicken embryos, which is depicted in the picture of some lab technicians that are inoculating eggs. But if we want to make a vaccine against a very serious pathogen, against a deadly virus or very dangerous virus, and we need to amplify a lot of this virus, this is very dangerous for the workers and for any spill out of the lab. So there must be very strict conditions, very high biosafety level to work with such viruses. So this is a very big disadvantage of this classical vaccine. When we want to use them against dangerous pathogen, we need to amplify this dangerous pathogen in a very high containment, in very good biosafety levels. One of the solution to this problem is production of new generation vaccines. They are genetic vaccines, and these genetic vaccines have been rapidly evolving in last years, and now everybody already heard about genetic vaccines because we have some new vaccines against COVID-19 that are genetic vaccines. So these vaccines are basically very different from the classical vaccines, as classical vaccines resembled virus particles that were somehow attenuated, which means weakened or killed. Here we don't introduce to our organism a ready viral particle, we just introduce the recipe, and it is our organism, our cells, that produce the vaccine on the basis of the recipe. So one of the methods to deliver this recipe to our cells is the use of genetic material itself. And in the vaccines that have been approved lately in the European Union against COVID-19, there are vaccines that consist of mRNA. This is messenger RNA, a type of RNA. And these RNA vaccines, when they are administered, they just deliver the recipe to our cells. Another type of delivery of the genetic material to our cells is the use of viral vector, which means that we use another virus, which is a more mild virus that will not harm us, and we use its natural ability to deliver its own genetic material to our cells, only that besides its own genetic material, we also introduce a part of the pathogen genetic material. So we introduce a gene encoding for vaccine protein into its genetic material, and then this virus delivers this gene encoding vaccine into our cells. To understand it better, we should remember what is the role of the DNA and RNA in our cells. So in our cells, the recipe for our proteins is present in the nucleus in the form of DNA. And the proteins are synthesized in the cytoplasm. So the DNA is in the nucleus, the proteins are constructed in the cytoplasm. So we need some molecule that will transfer the recipe from the nucleus to the cytoplasm. And this molecule is RNA. So first, information from the DNA is transcribed to the mRNA, messenger RNA, which then leaves the nucleus, goes into cytoplasm, and on the basis of the information that is written in the RNA, all the proteins are produced. So when RNA vaccines are produced in the vaccine factory, actually a similar process happens, but with crucial differences. So DNA is also transcribed into RNA, but it does not happen in the cell, it happens in the test tube in the laboratory. And then the RNA that is transcribed here, it is the vaccine itself. So like it is the vaccine preparation that is injected into our organism. And then the production of proteins on the basis of the information that is in the RNA, this process occurs in our cells and is governed by our cellular machinery. And the protein that is produced is a vaccine antigen, which means that this is the protein that resembles viral particles, which is to be learned by our immune system, and then similar protein on the virus will be recognized during infection. And viral vector vaccines, which, as I said, are also genetic vaccines, work in a little bit different manner. So by the means of genetic engineering, we obtain viruses that possess foreign gene in their genetic material. So they have their own recipe, and into this recipe we add a small fragment of the gene that encodes for a vaccine protein. And then this virus can naturally infect the cells, enter the cells, and deliver its own genetic material into cell nucleus. And then, together with its own genetic material, it delivers also the foreign gene, which encodes for the vaccine protein. And there are two vaccines based on virus vectors on the market in Europe right now. They are both based on adenovirus vectors. Adenoviruses are mild viruses that cause common mild infections like common colds. And these vectors have been already tested in over 600 clinical trials, so they were investigated very precisely. And to addition to the fact that these viruses are quite mild, and that is why they are safe, their vectors have been also manipulated, so they are replication defective. So even this mild virus, if it enters our cells, it is not so dangerous, but here it cannot even replicate because he is deficient with some of his own genes. And adenovirus vectors also have to be produced in cell cultures because they are actually viruses. So they need cells to propagate. So if we want to produce a lot of adenovirus vector, to have a lot of vaccines, we need to use cells. But because adenovirus vectors cannot propagate in our cells, we do not have to use so high containment, so high safety measures to produce these vaccines, because even if something happens, some contaminations, this vector is not as dangerous as a pathogen. So the basic benefits of genetic vaccines are that they are fast to enter clinical trials because they can be prepared very easily. Genetic engineering is now very highly developed, and it's easy to produce them, it's cheap, and also in terms of safety, it is much safer to work with genetic vaccine than with infectious pathogen. So most of the vaccines that are tested worldwide, and also all of the vaccines that have been approved in European Union, they deliver into our cells specific gene, and this is the gene that encodes, so it means that it has the information about the protein that is called spike. And spike protein is the surface protein that is on the surface of the virus, and it acts as a key that binds to the lock on the cell. So it binds to the receptor, enabling the virus to enter the cells, and it is also the main target of neutralizing antibodies. So the antibodies that are produced by our immune system and that can bind to this spike protein and block it from binding to receptor and entering the cell. So you can see here how this anti-spike immunity works. So first we deliver a spike protein in form of the vaccine. So we can deliver either the spike itself as a classical vaccine, or as in approved vaccines anti-COVID-19, we can deliver recipe for the spike, and then the spike is produced by our cells. Then our immune system produces antibodies that can recognize the spike. And then after weeks or months, when the real pathogen enters and it presents spike's protein on its surface, the antibodies are able to recognize it. They bind to the virus and they block the virus from entering the cells. And here you can see the vaccines that have been registered in European Union and some other countries. So basically here we have two mRNA vaccines, which is the Pfizer vaccine COVID-19 and Moderna vaccine. And also we have two vaccines based on adenovirus vectors, and this is Oxford vaccine and one-shot Johnson & Johnson vaccine. Also in other countries of the world there are different types of vaccines that have been registered. So besides genetic vaccines, that are also some classical vaccines that have been registered, like vaccines based on inactivated virus, which means on a killed virus, or also subunit vaccines, which means that these are just parts of the virus, portions of the virus. So one of the questions that I hear a lot of times is why SARS-CoV-2 vaccine was developed so fast. So why cannot we develop other vaccines as fast as SARS-CoV-2 vaccine? Why don't we have a vaccine against HIV and HCV? So first of all, I would not like to compare vaccine development for two different pathogens, because there is just molecular biology of pathogen is so different that it is very difficult to develop vaccine against hepatitis C virus or HIV virus because of their specific molecular properties. But what happened that the COVID-19 vaccine was developed so fast? So first of all, as I already told you, there was a really rapid development in genetic engineering in last years. And for example, if you compare our ability to sequence virus genetic material, so to decipher the information in the virus recipe, when there was epidemic on the SARS-1 virus in 2002, it was several months to decipher the genetic material of the virus, whereas now with SARS-CoV-2 we needed only like 2 weeks to decipher the same information. And what is important, if we want to develop genetic vaccines, we just need to know the genetic sequence of the pathogen. So we just need to know this recipe to start production of the vaccine, to start testing of the vaccine. So we do not need to have the infectious virus at all. We do not need to have it in our lab. It's only enough to get this information by email. So this is just the sequence of GTCA letters like GTT, CC, ATC. And on the basis of this, we can design and start producing and testing the vaccines, because it's only information about genetic sequence that we need. Also genetic vaccines are fast and cheap to produce, and also a pandemic regime was very helpful, which means that the testing and also the regulatory approval of the vaccine was shortened, and I will show you in the next slide how that was done. Also, the research on the vaccine was really massive. There were from the beginning over 90 vaccines in trials, and there was a lot of additional financing, and also, but not last but not least, because it was very, very important, that we had already promising results for SARS-CoV-1 vaccine development. And actually scientists really used a lot of information from the research on SARS-CoV-2. So basically, what they did was to use the vaccines that were the most promising for SARS-CoV-1, and they just exchanged the gene for SARS-CoV-1 in some cases, and it worked. So they knew what gene we should take. So I want to tell you also about how vaccines are tested, so that we can understand better this whole process. So first of all, we have the first stage, which is research and development, which serves us to choose what antigen we want to use, so like what part of the virus we want to expose to our immune system. Then we need to optimize the procedures to obtain this antigen, to purify it from other contaminating molecules. So this is usually a very long phase. Then there is a preclinical research, which means that we inject the vaccine to animals, and then we analyze the immune responses, so how it boosted the responses in the animals. And also for some viruses, if we have good animal models, we can check if this is protective, if it protects against the infection. And then we go to clinical studies, which means that we start studies on humans. And then clinical studies are also divided in some stages. So we have phase 1, when we have just few people and we check if the vaccine is safe. Then we have phase 2, when we have more people, we still check for safety, and also we analyze immunogenicity, which means that we analyze how immune responses are boosted after the vaccine. And then we have the phase 3, which is a randomized trial, which means that we have two separate groups. One group receives the vaccine and one group receives placebo. And then after some time, we compare statistically the number of infections in both groups, so we can calculate if there is statistical difference between the groups and if the vaccine was protective. So here you can see how this vaccination development was performed during COVID-19 pandemic in this shorter regime, let's say. So first phase, as you remember, was research and development, which is design and optimization of production of the vaccine preparation. And here, instead of the years, it was reduced to almost nothing, because as I told you already, in many cases researchers just took the same approach as was already tested for SARS-CoV-1. So then when they received the genetic sequence of the SARS-CoV-2 virus, they just took the same gene, the same portion of the gene, and they put in the vector, so in the virus vector, or they put it as an mRNA. So then the preclinical testing in animals was actually done in majority in parallel with the clinical trials. And then also, as you can see, the different phases of clinical trials were overlapping. So the phase 2 started before the phase 1 was finished. Only after they obtained some initial data, they already could start phase 2, and the same for phase 3. And also, what is important, already during phase 3 clinical development, the production of the vaccine was started at risk. So it means that even though the producer didn't know if the vaccine would be approved, they started to produce the preparations anyway, so that after approval they could be administered, distributed widely worldwide. So that is why it was so rapid and so fast. Also, here you can see how the numbers of the COVID-19 development. So you can see how many trials were done preclinically. So now it should be even more, because this is not the newest graphic. So like there were hundreds of vaccines tested. From them, tens went into clinical trials, and from them we have several vaccines worldwide and 4 of them in European Union. So you can see how massive this research effort was. So now, as the vaccination is already quite widespread and many of us have been vaccinated, there is fear about the vaccine efficiency because there are some new viral variants that emerge, and people are not sure if the vaccine will work against this variant or not. So I wanted to give you just a short information about these virus variants. So basically all viruses mutate naturally, so it is completely normal that the viruses mutate. And this happens because the replication of the viral genetic material in the cell is error prone. So it means that there are some errors that make the changes in the genetic material. And because they are just random errors, most of these new viruses will be less fit than the non-mutated virus. But some viruses randomly will acquire mutations that will be beneficial for the virus fitness, and then these variants will be able to spread more easily and will be able to become dominant in the society. So SARS-CoV-2 virus is not as variable as some other viruses like influenza virus or HIV virus, but we can observe some rapid emergence of variants lately. So why is that? So the main reason for this is the rapid global spread of the virus. So the global rate of replication of the virus genetic material is very, very high. So that means that even if the errors during the replication occur not so often, but because this rate is so high, this error rate will also be higher. So there will be statistically some variants from time to time that will benefit from such error, and then the new variant can dominate in some region, and then it can spread to the whole world. So medical agencies of the world determine some variants that they call variants of concern. So they are variants for which we have the proof that they have higher virus transmission or virulence or escape from immunity, which means that even after vaccination or after getting one infection, we can still get infected with this virus because it would escape developed immune responses. There are also other variants that are called variants of interest, and these are variants that have mutations, that have changes in some part of genetic material that we think that can cause some trouble, but we don't have proof yet. So, for example, there are mutations in the genes encoding for spike protein, which is used in the vaccines against SARS-CoV-2, but we have no data about higher transmission, virulence, or escape from immunity. Because some new variants of SARS-CoV-2 have higher spread, this is quite serious for our societies, because then we can have more infected individuals, and that can lead to overburdening of our health system, so there might be not enough places in the hospital and so on. And if the vaccine efficiency is somehow influenced by the new virus variant, that also can be problematic. So these are like the two major concerns that are concerning the new variants. So what can we do? So first of all, what researchers do is the variant monitoring. So we need to know which variants appear in what region, if they spread or they do not spread. So this is very important. Then another field of interest is vaccine testing. So we want to know which vaccines are efficient against which variant. And until now, I want to underline this. There is no significant drop in efficiency of the vaccine against severe COVID-19. So we have some drop of efficiency for some vaccines against some variants, but generally there is still effectiveness against these variants. So even if there is a drop, it is a slight one. And also the vaccines still protect against the severe COVID-19, against hospitalization and death. Also, what scientists check is the probability of reinfection with the new variant, but still we do not have data if it is possible to reinfect with new variants more easily. So I would like to conclude that the most important thing for now, I think, is to limit virus replication globally. So because the virus replication is so high, the virus is still transmitted very potently, and this is the reason for a new variant emergence. So we need to do what we can to vaccinate the population of the world the most we can, most efficiently. So then this can limit virus replication and variant emergence. I would say that this is the most crucial task right now. And with this, I would like to thank you from the University of Gdańsk, which is situated in Tri-City. Tri-City consists of 3 different cities: Gdańsk, Sopot, and Gdynia, each one with its own character, and they are neighboring cities, very close to each other. So they're like one big city with different spots, with different character in each of the cities. So I invite you very much to see these cities by your own. Thank you very much.